Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. The views and opinions expressed by our guests are their own and do not necessarily reflect the views of the host, the American Phytopathological Society, or any affiliated entity. This is the 6th episode of 2026, and the first of a 3-part series featuring 2026 APS Fellows. Today I welcome Dr. Christina Cowger, Research Plant Pathologist with the USDA-ARS Plant Science Research Group, and full adjunct professor of plant pathology in the Department of Plant Pathology at North Carolina State University in Raleigh, North Carolina. Dr. Cowger completed a BA at the University of Oregon and an MS and PhD in plant pathology from Oregon State University in Corvallis. Before being hired as a research plant pathologist at USDA-ARS in 2003, she served as a postdoctoral fellow at OSU. Dr. Cowger is an internationally recognized expert on cereal diseases who has made diverse and original contributions to epidemiological and applied knowledge of wheat and barley diseases and their management. Dr. Cowger's federal research program focuses around 3 important diseases of cereal crops, including powdery mildew of wheat, fusarium head blight, and Septoria nodorum blotch. She has a strong commitment to translational plant pathology, and her leadership has brought together researchers, crop advisors, and grain producers in efforts to increase the use of resistant cultivars, many of which she helped to develop over her career. Dr. Cowger is a committed mentor, having supervised 10 PhD and master's students in her affiliation with North Carolina State University. Dr. Cowger has been involved with APS since the late 1990s, serving as associate or senior editor for 4 different APS journals. She has also served on the nominations committee and the scientific program board. Welcome, Dr. Cowger.
Thank you very much for having me.
I want to first congratulate you on being elected a 2026 APS Fellow. That has to feel pretty good. Tell me how that went down, how you were notified.
I got a call from Carolee Bull, the president. It was a little bit out of the blue. I was very thrilled and also very humbled because I'm in the company of so many excellent plant pathologists, and it feels great, but also, like I say, humbling to try to be as good as an APS fellow should be.
Yeah, I think they're pretty good at picking the right people, and certainly there's people that probably could have received them in the past and didn't, but typically if you're receiving one, you stand out even among those. Congrats again. As a reminder to our listeners, APS bestows the honor of fellow to current APS members in recognition of significant contributions to plant pathology and or the society in one or more of the following areas. It could be in original research, it could be in teaching, it could be in administration, it could be in public service, or extension and outreach, or any combination thereof. Now, Dr. Cowger, you certainly wear a lot of hats, but being an employee of the USDA-ARS, I assume that some of that recognition is for public service. Is that correct?
I think so. I've tried to form my career around the goals of sustainability and service to the grower community, the wheat and barley industries writ large, but particularly starting with producers. So I see my job as trying to help people grow wheat and barley more profitably and more sustainably. And from that flow a lot of other priorities about what to do. But yeah, I think my work has been mainly in the areas of disease resistance, understanding the epidemiology of diseases in order to better manage them, and really breeding for durable resistance.
And certainly you have a really impressive CV and research outputs. So your original research is a big part of that, of course. And I feel like with a lot of fellows, it's like you can check off every box. I understand as a USDA-ARS employee, you do some adjunct teaching, and the teaching is guest lectures and things like that. So maybe less in terms of the teaching bucket, but all the other ones, original research, administration, public service, extension outreach, you encompass all of that.
Yeah, I think that's true. And over the years, we've focused on certain diseases that are economically a priority for growers in the United States, as well as in other countries. And we've done some basic research, but a lot of it has been in function of trying to understand how resistance on the part of crop species works, and therefore how to deploy it more efficiently.
Yeah, I think it's always nice when you work in a system that's so vital to our food systems. Wheat and barley and cereal crops are so important to our survival. So to be able to advance research in those areas to have more disease resistance or to combat certain pathogens is something that I think a lot of people can get behind. It's very intuitive why it's important. You've been pretty active with APS, coming back to this, giving them service and giving them your time and you being recognized for your research and all these things. But certainly you've been really active with APS since late 90s, early 2000s. You've served as associate editor for several of their journals: Phytopathology, Plant Health Progress, Plant Disease, Phytofrontiers. You've also served in various other capacities in terms of nominations committee and the science program board, section chair. When did you first get involved with APS, and what's your earliest memory of a meeting that it doesn't have to be your first meeting, but your earliest meeting that made you realize, wow, this is a really worthwhile organization?
Yeah, that would have been in, like you say, the late 90s or early aughts. And I, as a graduate student and then a postdoc, became aware that APS was where a lot of other people could be found who would give insight into the things I was finding and also connect me with ways to spread my research more broadly. I helped to organize an early symposium at APS that had to do with agriculture and its vulnerability to attack. This was shortly after 9/11. It was a very interesting time to be thinking and talking about where agriculture fit into that context. And so I thought it was a valuable contribution to bring together some of the best minds in our science to talk about that.
Certainly, and that's an important topic. One of the things that stands out in your volunteer work with APS is serving as all these associate editors and senior editors. What makes you so interested in serving in that role of editor? Because not everybody can do it, but I find that the people that do it really like it. I've served as an associate editor and things like that, but I'm curious, what's your take on it?
I would say I had a really good role model in my graduate advisor, Christopher Mond at Oregon State University. And Chris was all about service to APS and was really good at explaining the peer review system and modeling how it should work. And so I came to realize that peer review, if it's done well, is really at the heart of science because it's a way that you present your ideas as best as possible and then receive critical feedback on them from anonymous reviewers. And 9 times out of 10, that ends up strengthening the paper a great deal and strengthening your thinking about your own results. So for me, it's not just editing or reviewing, it's really an integral part of optimizing scientific achievement and communication.
Sure. And thinking back to my earlier career, not that I'm like at the end of my career, but just thinking back to earlier when I was a grad student and postdoc and assistant professor, you'd receive these reviews back and you take them so personally. It was like, why are they ripping me apart like this? And then you realized as you started to do it yourself, wow, they really took the time and care to go through this and to help strengthen this. And I like seeing early career scientists figure that out. Receiving these really extensive reviews is the reviewer really giving their time to you, and it's really a nice gift.
That's absolutely right. And of course, it's important to do it in a kind and generous way, but you can be kind and generous and honest also. It's just all in how you say it. And I agree. I think it's one of the ways that junior scientists mature and come to understand what's needed in order to get their ideas across.
Yeah, I agree. And the other thing that I think that junior scientists still get out of it today, even for me as a mid-career scientist, is that the more papers you review and read that are even outside your expertise, the more well-rounded of a scientist you are, and you get really good ideas. Oh, wow, that's a really cool technique. Maybe we could apply that to our patho system and uncover some really interesting things about it like they did.
Yep, I think that's true. I think also that if you're an editor, it's incumbent on you to get reviews for the people who submitted the manuscript that are coming from different directions, and get reviews that are thorough and really get at the heart of what the manuscript has to contribute. And all of that takes a little bit of experience to try to figure out who's going to be able to give that kind of review, especially if, as you say, it's not directly in your own wheelhouse. You have to do a little bit of research to figure out what kind of review is needed for this
Yeah.
manuscript.
Now, you're an internationally recognized expert on cereal diseases who's made significant contributions to our understanding of wheat and barley diseases. What got you interested in cereal diseases, or when did you first get on the path of studying cereal diseases?
That was for my master's program, again, at Oregon State University with Chris Mont as my advisor. And Chris is a researcher of cereal diseases. And as I thought about it, wheat, barley, essential to our diets, essential to our economy, something that I could feel good about working on for the rest of my career. And we have to have wheat to survive, to eat. And there are a bunch of different ways that human beings use wheat and also barley for that matter. So it's been about 25 years. No, it's been about 30 years, and I'm not tired of working on those crops yet. And of course, working with USDA, you're hired to contribute in a particular crop area usually. And so that jibes well with my attitude toward my work, which is trying to benefit a particular industry, a particular set of producers, trying to make conditions more profitable, trying to improve sustainability so that fungicides are used judiciously and not overused and minimized to the extent possible, for example. So yeah, I have that kind of crop-oriented attitude toward my work.
Yeah. Now, I read that the United States ranks fourth in wheat production based on 2024 numbers, producing some 54 million tons, 21 million tons of which are exported. And U.S. is third largest exporter of wheat behind Russia and Canada. Now, as I dig into some of your work, I see reference to different wheat varieties, soft red winter wheat. I have to be honest, I think I'm like a lot of people. I don't understand the differences in the types of wheat we grow commercially here in the United States. Could you give us a basic lesson in the types of wheat that are produced in the US and used domestically and even exported?
Yes, sure. There are several market classes, I guess you could say 5 basic market classes of wheat in the United States. The first being winter wheat, and that's the largest. That's broken down into hard red winter wheat and soft red winter wheat. So hard red is grown in the Great Plains of the United States, and hard refers to its protein content. It's appropriate for bread making, and that's about 40% of total U.S. production, hard red winter wheat. And then soft red winter wheat, which is grown essentially east of the Mississippi River, that has a lower protein content,
Okay.
and it's used for cookies, crackers, pastries, and predominantly also in the Southeast for animal food. So it's an essential component of livestock production. Then you move up north in the Great Plains and you find hard red spring wheat, which is about 25% of all of our wheat production in the U.S. And that's higher protein as well. And that goes into specialty breads and then blending with lower protein wheat as necessary to hit the right protein targets.
Okay.
Growers make, I think, around 10 bushels an acre more in production from winter wheat than spring wheat. And so those who can grow winter wheat do, of course, but as you progress north, sometimes the winters become too harsh for winter wheat, and so you have to wait and plant in the spring, and hence that's where spring wheat comes from.
Could I ask a dumb question? Winter wheat presumably is planted in the fall. Is that right? Oh,
Yes,
and when is it harvested?
it is harvested anywhere from April to June, depending on your latitude. So the further south you go, the earlier it will be harvested, and the further north, the later,
Okay.
because you need a longer growing season. And then spring wheat would be planted in, say, May usually and harvested in August or early
Yeah.
September.
Typical growing season crop when we think of spring wheat, but winter wheat is a little bit different.
Yes, correct. Yep. And then white wheat is grown in the Pacific Northwest mainly. That's about 15% of our total wheat production. And that actually has both winter and spring habits, depending on how cold your environment is again. And that, a lot of it goes to the Asian noodle market. So that's,
Oh, really?
yes, a big consumer of Northwest wheat is sent to Asia. And actually, that component has been increasing. Thailand, the Philippines, Korea, Japan consume a lot of U.S. wheat. It's also used for crackers and cereals. Finally, you get into durum wheat, and that's only about 3 to 6% of total production, but it occupies an essential role because it's the only wheat that's really appropriate for the pasta that we eat here in the U.S. And of course, that people in Europe also produce and eat.
We eat a lot of pasta. It seems like that's a small percentage of total wheat production, given the amount of pasta consumption probably here.
That's true. And it's possible that we acquire durum wheat from across the border in Canada because these northerly regions are located right across the border into Canada. So durum production is also large scale in the plains provinces of Canada.
Okay, so the bread wheat is the hard red wheat, winter wheat, right?
That's correct.
Would you say that represents the largest percent of wheat production domestically?
Yes, yes. Hard red winter wheat grown in, say, Nebraska on north to southern Minnesota and the Dakotas. That constitutes about 40% of total U.S. wheat production.
Okay. Well, thank you for that really nice overview of wheat production in the US because I want to talk about the different diseases you work on, but knowing these different market classes is really useful in coming back and referencing them. You've devoted a lot of your career to different diseases of wheat: powdery mildew, fusarium head blight, Septoria nodorum blotch. When I look at your CV, you've tallied some 30 plus publications on powdery mildew, 20 plus on fusarium head blight, 15 or more on Septoria nodorum blotch. These are just estimates, but which of these three that you've worked on, and certainly you've given a lot of your time and energy to these different pathosystems, diseases, which of these are the most economically important diseases of wheat today?
That's a tough question, actually, because it depends a lot on your environment. So in the eastern U.S., I would say without a doubt it's fusarium head blight year in and year out, because oftentimes conditions in the spring when wheat is heading and flowering and it's therefore vulnerable to fusarium infection, oftentimes those conditions are wet enough to make it a severe year for fusarium infection.
For spring wheat. So winter wheat you don't deal with fusarium head blight in winter wheat?
No, you do. You do.
Okay.
Either spring wheat or winter wheat is vulnerable potentially to fusarium head blight. And the window of infection for either one is just basically, it's determined by the weather about 2 weeks ahead of flowering. So if you get a rainy period 2 weeks ahead of wheat flowering, you're susceptible to a bad fusarium blight epidemic. Yeah.
As we know with fusarium, we're dealing with mycotoxin production. And presumably there are thresholds at which whole batches of wheat are rejected based on those. Do you want to talk a little bit about how that works?
So that is correct. The contamination of the grain, and this includes wheat and barley, with the mycotoxin deoxynivalenol, or DON for short, is one of the worst consequences of Fusarium head blight. And when you get this toxin in the grain, which is sometimes called vomitoxin, even a low concentration can render the crop unsuitable for human consumption. So up to 2 parts per million of vomitoxin is usually allowed in wheat that's destined for human consumption, and generally then up to 5 parts per million for animals, sometimes a little higher. Swine are particularly susceptible to poisoning with vomitoxin. And that means that in a bad Fusarium head blight year, the elevators, the mills that are purchasing grain from farmers, are going to be testing each load to see what the vomitoxin concentration is. So that can really wipe farmers in whole regions out. Usually they can then turn around and sell it at a discounted price for animal food, but they have to drive farther, so it's uneconomical to transport the crop, and it can be a disaster.
You've probably heard the expression, not that it's a great one, but the solution to pollution is dilution. And I wonder, when you have a rejected load, if that is just mixed with one that doesn't have any, just to not have to throw away all that, just to lower the amount and make it acceptable for use.
That can happen easily, especially in a bad year for Fusarium head blight. The farmer will receive a discounted price, and then the mill will have to figure out how to mix loads to achieve a lower level of deoxynivalenol or vomitoxin in all of the wheat that it generates. That is usually costly. So everybody loses financially when there's a bad Fusarium head blight or scab epidemic.
Yeah. And just to remind the listeners, I think this is a really important distinction here. As plant pathologists, we all tend to work on different pathogen groups that cause loss of yield. And obviously that could be financially devastating to a farmer. But then you have the added mycotoxin production, which means that the yield that you did get could also be rejected. So it's like a 1-2 punch in terms of you've lost some yield and the yield you got may be rejected outright. Is that true of so you work with powdery mildew and Septoria nodorum blotch? Those don't have any toxin productions associated with them. Presumably powdery mildew doesn't. I don't know much about the blotch disease you work on.
No, you're right. Neither of those diseases results in a mycotoxin in the crop. The main losses for powdery mildew have to do with just lowered photosynthesis on the part of the wheat plant, because to the extent that the leaves are covered with lesions, they can't photosynthesize. The powdery mildew is an obligate pathogen, and so it's hanging out on the leaf, nourishing itself on the photosynthate produced by the wheat plant. And consequently, you get fewer tillers, which are the stems that produce heads.
Yeah.
And you also get grain that is less well filled out. It's more shriveled.
Okay. And because I like to always educate our listeners, you said obligate, and we're talking about an obligate biotroph. And what we basically mean by that is that this pathogen keeps its host alive to steal from it, but not kill it, or at least kills it very slowly, so that maybe the plant is harvested before it's killed. Because if the plant is killed, the pathogen is killed. It relies on it to complete its own life cycle.
Correct. Yes. The wheat powdery mildew generates these little, what they call green islands around its lesions. So if the whole rest of the leaf is turning yellow, brown, is senescing, we call it, the little green island around the mildew is still blithely producing the photosynthate for the pathogen that, as you say, has commandeered those cells.
Yeah, absolutely. Now, a lot of your publications and even the description of your research on the APS Fellow website talks about breeding for resistance. Do we have multiple resistant wheat? Is it called lines, wheat lines, against these different diseases you work on? And in terms of breeding, where have we made the most progress and where do we still have the most problems?
I think both powdery mildew and Fusarium head blight are pretty good examples of breeding progress. Both of those diseases are significant problem in the eastern United States. Taking powdery mildew first, the problem that growers face is that if we deploy a particular resistance gene widely, then we put selective pressure on the powdery mildew pathogen population to overcome that resistance gene. And that's happened several times in the last couple of decades where farmers have used a particular resistance gene widely in their varieties. They think, oh, great, this one doesn't get powdery mildew. So they plant many hundreds, thousands of acres of it, and pretty soon that gene goes down and doesn't work anymore. So breeders have had to continuously identify new sources of powdery mildew resistance, and they've turned to wild wheat relatives, which there are many of in the Middle East, for example, grassy plants that share genetics in common with wheat, and they've introgressed or moved those wild sources of resistance into adapted wheat backgrounds. And so a lot of what pathologists have been useful in doing is helping those breeders identify when a resistance gene is genuinely novel, when it's really new and can be thought of as a different resistance gene, because what we try to do is not just have one or two resistance genes to mildew in a wheat background, but have several of them so that less selective pressure to overcome a gene is exerted on any one of those genes.
Yeah, that makes sense. It's the way we think about mode of action. You don't want to have different modes of action for antibiotics or antifungals, because if you're using different modes of action, then you can't overcome it, especially if you're alternating between one mode of action and another. You're keeping it from developing a workaround.
Yeah, exactly.
Now, when you're breeding for resistance for a certain, let's just say a powdery mildew, are you starting off with a background that already has Fusarium head blight resistance? Are you breeding for resistance against multiple diseases, or the varieties that are used differentially susceptible to those? And you focus on Fusarium head blight for the soft winter wheat and powdery mildew for something else?
Yeah, I think what you're getting at is breeders can only tackle so many diseases in a given wheat line, right?
Yeah.
Wheat gets a lot of diseases and a few insect problems we haven't really talked about yet either. A breeder for a particular geographic region has to focus on the main problems in that region. I think what's been really positive in the eastern U.S. is that over the last 25 years or so, a lot of breeders have managed to put backgrounds of resistance into their germplasm pool out of which they draw their commercial lines that are broadly resistant to powdery mildew, for example. You don't see as many commercial varieties anymore in the official variety trials in the eastern U.S. that are susceptible to powdery mildew, not badly susceptible. They might get a little bit of mildew, but that's okay. That's owing to the efforts of breeders to broaden that sort of quantitative resistance background that's present widely in the germplasm.
And also, just to think more general about plant pathology, we always talk about the three components of the disease triangle being the susceptible host, the virulent pathogen, and the favorable environment. But not all pathogens have the same environment that makes it favorable for them. So some require a cool, wet spring, some require a scalding hot summer. You're not necessarily going to always have all the conditions that are going to be suitable for all the pathogens in a single growing season.
That's exactly right. There are a few problems that tend to be widespread across the U.S., barley yellow dwarf virus being one of them. So any advances that we make on that front could be applicable even in, say, the Rocky Mountains or the Great Plains. So, in fact, recently, Paul Murphy, a breeder at North Carolina State University, did manage to introgress a gene for resistance to barley yellow dwarf virus into adapted germplasm, and it's now in the process of making its way into commercial varieties. And we were able to show in a field experiment over multiple years, working with some entomologists here, how very effective this barley yellow dwarf virus resistance gene is. So it's not acting against the aphid, which vectors or transmits that virus into the wheat plant, but rather it's doing something inside that wheat plant that is allowing it to repress the virus. And the effect is quite dramatic, actually, very encouraging. We're publishing some research, hopefully soon, in an APS journal that will demonstrate to people just how effective this barley yellow dwarf virus resistance gene is, even in the absence of very intense disease pressure.
Yeah. We talked about the three big diseases you've worked on, and now we're talking about barley yellow dwarf virus. Of course, there's other very notable diseases of wheat. Like you said, cereals get a lot of diseases, but I'm thinking of wheat blast, wheat stem rust, Septoria triceti blotch. How much of a concern are those diseases in North America? And if not, where are they the most problematic?
Wheat blast is primarily a problem in Latin America, and there has, over the last 30 years or so, been serious concern about whether it might migrate to the United States. But so far, so good. We haven't really seen wheat blast take hold here. Let's see. Stem rust is a huge problem in Eastern Africa and across the southern part of Asia. And that is owing to new mutants of the stem rust fungal pathogen that have overcome resistance genes in wheat that were historically effective against it.
Does that include UG99?
Yeah, with UG99 being the prime example, and then some further mutations from the UG99 mutant. So again, hasn't shown up in the US. Conditions here may not be favorable for it. And we have a little bit of stem rust in the far southern part of the country, but it's nothing that's economically important.
And that would fall under the role of USDA-ARS in terms of monitoring for that being brought in UG99.
Correct. Yes, that's right. There are two wheat rusts that are present in the U.S. and can be a significant problem. One of them is stripe rust, and that's particularly problematic in the South Central states and the Pacific Northwest. And then leaf rust is ubiquitous in the eastern U.S., usually not a serious economic problem unless the variety is particularly susceptible.
Okay. One thing you had mentioned, we were talking a little bit about disease vectors. Pests and disease have been a concern regarding wheat for a long time, going all the way back to Thomas Jefferson. I've read recently about a little bit about the Hessian fly or the barley midge in the late 18th century causing problems. And just in preparing for our conversation today, I was doing some reading and saw that the wheat stem sawfly is a chronic pest of wheat in North America. Is the barley midge still an issue? And what kind of impact do those two, or even wheat stem sawfly, have on wheat production?
Hessian fly, as you say, was introduced probably in the late 18th century. It remains a serious problem in the southeastern United States and a little bit of the Great Lakes soft wheat area. And it is analogous to powdery mildew in the sense that many genes have been brought into wheat to provide resistance to Hessian fly. But as soon as one of those genes becomes widely deployed, it puts enough selection pressure on the Hessian fly population that it often has been overcome, particularly maybe in certain states. So there's a constant effort to identify and bring in new Hessian fly resistance genes. Here in North Carolina, a novel Hessian fly resistance gene has been discovered that is partially successful. So it imparts quantitative resistance, and that is seen as an advance in the sense that it reduces that selective pressure on the pathogen. It can reduce the amount of pathogen damage without killing the Hessian fly outright. And because it's only partially effective, the Hessian fly population is less selected to overcome it.
Is ergot still a problem at all, or is that more in just a small batch organic rye fields in upstate New York?
Ergot is a problem, I believe, in the upper plains, maybe in Michigan, Minnesota, the Dakotas. It's not their number one disease problem, but it can rear its head for sure.
Yeah. For those of you that aren't familiar with ergot, this is Claviceps purpureae, and this is the alleged pathogen, although I think it's been debunked, that was associated with the Salem witch trials because the fungus produces lysergic acid, very similar structurally to LSD, and causes some interesting side effects. But it's obviously another one of these contaminants. It it lowers yields, but it also gets introduced and produces its own mycotoxins.
Correct.
Yeah.
Yes.
So your work generally seems to fit under this large umbrella of molecular plant pathology. Could you tell us a little bit about some of the tools that you've developed and use in your lab to answer some of the pressing questions regarding cereal diseases? As a follow up to that, I recently read your, not end to end, but I looked over your ISME communication paper on synthetic spike in metabarcoding for resolving fusarium species and associated mycotoxins. Really interesting paper. Do you want to talk a little bit about the tools you use to approach some of the pressing questions in your research program?
Sure. The one you're talking about, synthetic spike in metabarcoding, it responds to two problems. One is that although Fusarium graminearum is the most common species infecting wheat heads and barley heads and contaminating them, there are other Fusarium species and they produce other mycotoxins which are not DON or vomitoxin, but rather sometimes things called moniliformin or eniatin. These are more common in Europe, but they can show up here. We have not actually monitored them very well, but we have shown in some of our work that those emerging mycotoxin producers, as they're called, do occur in wheat heads in the eastern US and apparently also in Kentucky. So we're still working on identifying, okay, where are these emerging mycotoxins likely to be a problem so that we could monitor for them. Meanwhile, one of the ways that we're doing that is by sampling symptomatic wheat heads broadly across many wheat fields and then identifying the Fusarium species that are in them. Now,
Right.
it has been up until now the case that you needed specific primers. You needed to know which species of Fusarium you were trying to amplify, right?
Good luck with that.
Yeah, Fusarium has a million species. So the advance in this metabarcoding paper, which our graduate student Pete Oppenheimer has made, is that you can amplify without specific primers a certain stretch of the TEF gene, which is broadly present across all the Fusaria. You can sequence it and use an a library of Fusarium sequences to identify it to species
Okay.
without having had the specific primer. So it's what's called species agnostic in the sense that you don't have to know the species ahead of time. You can find it out through this test.
That's good given how many novel unnamed species there are in Fusarium across the several dozen species complexes.
No, it's very broad. And not only can you do that, but you can also, that allows you to, in turn, predict what mycotoxins might be present that you maybe should be testing for, right?
Yeah.
And you weren't. And then the other thing that you can do using this technique, the spike in is this piece of DNA that you put in all your samples in the same quantity. And so regardless of whether there are differences in the strength of your amplification, you know how much spike in you had in each sample. And so you can use that to control how much of the Fusarium DNA and therefore biomass you deem to be present in each of your samples.
Okay. Yeah, that's really innovative.
It's basically a reference, right? So
Yeah.
I can then determine if I have multiple wheat heads that are all symptomatic, I can find out which Fusarium species were in each of them and how much. I can estimate the biomass of each species in each head.
That's awesome. And just thinking, tying this back to your kind of translational research and breeding, is that you're breeding for resistance against Fusarium head blight, right? Presumably, some Fusaria, which may actually produce novel mycotoxins that are yet to be characterized, you may be increasing populations of those. So there's this whole, obviously, you're taking out the vomitoxin producing Fusarium, which is very important for food security, food safety, feed safety. But you bring up this larger point that there's still a lot of unknown fusarium species with unknown mycotoxins that could potentially emerge in the presence of a resistant cultivar of wheat to fusarium head blight, no?
Yes, it's an extremely complicated picture because then you have to factor in the environment also.
Yeah,
So we are hypothesizing that the reason Kentucky, for example, appears to have a lot of these emerging mycotoxin producers may have something to do with how much grassland and pasture land Kentucky has in comparison to, say, an East Coast state, which has much more row crop land. So we may not have as much of these emerging mycotoxin producers. It may have something to do with the crops that are grown around the fusarium population. Fusarium also spends a part of its life every year on something else.
Yep.
So you got to factor that in too. Maybe it's on corn,
right.
maybe it's on some grass. So there are many factors that could lead to variability in the fusarium species that are common. Even in a given field, it may vary from year to year depending on what the rotation crops are.
Sure. Before we wrap this up, I want to pivot a little bit and talk about mentoring because it seems like something that, based on your APS fellow profile, mentoring is, like, to a lot of scientists, mentoring is very important. And over your career, you've supervised 10 PhD and master's students. You served on advisory committees or as a mentor for dozens of other graduate students, postdoctoral fellows. You've had a lot of visiting scholars from different countries, probably in your capacity with USDA to host those. Do you want to tell us a little bit about your views on mentoring? And you've taught some too. You give guest lectures. Do you like the more formal classroom teaching, or do you like the more one-on-one or small group mentoring?
I like both really for different reasons. The guest lecturing that I've had the opportunity to do is in a course called Breeding for Disease Resistance that is offered here to graduate students at NC State. And what that does that I think is helpful is allow students to understand how disease resistance is actually deployed in real commercial crops. And then what impact does that have on the pathogen population that is attacking those crops? And that part of it is mine to present, which is what do we know about the way that hosts and pathogens as populations interact in wild settings? And how does that differ when you have an agricultural crop that you're manipulating? What impact does that have on the pathogen populations and how do they respond? So it's a lot of population genetics. It's some things about management like host heterogeneity, which is mixing different cultivars of a certain crop within a certain field. It's giving people a window into the practical implications of using disease resistance.
But it speaks to the value of having these relationships between universities and USDA-ARS, is that you have all this expertise that, like, you're dealing with real world problems on really important crops that are used both domestically and globally. And you can give them real life examples of, hey, just this year we introduced a new cultivar against this, and let's see how it works out.
Exactly. And there are many examples from the United States, but also from other countries in the world to help people really understand, okay, what happened when they planted willow trees in Northern Ireland in grids of different varieties? Did that reduce the willow rust or not?
Yeah.
So those are interesting and useful for people who are going to be spending their lives trying to deal with commercial disease problems. Mentoring of individual graduate students is also important. And I'm sure how rewarding it is to see a graduate student who comes in and is halting and hesitant in the lab, but then develops skills over time that allow him or her to be an effective bench researcher and to really strategize about what experiment they need to do next in order to advance the understanding of where a particular gene is located or whatever.
Yeah. Yeah, it's been really rewarding in terms of having even undergrads in the lab that eventually realize they want to pursue grad school and they stay on. I've had a number of undergrads that stayed on and did a master's or a PhD with me, and it's so rewarding to watch them really develop over their early career from being that kind of hesitant, insecure person to this really confident, well-spoken early career scientist. It's been a nice experience for me, having mentored over a dozen grad students over the last 10 years for me.
Yeah, it's great, as you say. And right now we're having the experience of a graduate student mentoring a younger grad student, both of them mentoring an undergrad
Yeah.
for summer. And it's really cool to see knowledge being imparted through the pipeline like that.
Yeah, and I love that, having that, because that peer mentorship is really important too, because students feel less dumb asking a question, even though it's not a dumb question. Like you said earlier, it's no dumb question. They feel dumb asking someone with a PhD. So if they can ask another student, they feel more confident in that. But it's been really great speaking with you today, and I appreciate you taking the time. And again, congrats on being named a 2026 APS Fellow.
Thank you so much, Matt. I am honored that you would interview me. Thanks for doing it. And I've really enjoyed talking with you.
Absolutely. You've just heard from Dr. Christina Cowger, research plant pathologist with USDA-ARS in Raleigh, North Carolina. Be sure to check out the show notes for links to Dr. Cowger's APS Fellow profile and her research program. Lastly, I'd like to acknowledge America's semi-quincentennial, which is this month. Happy 4th and be safe. I'm Matt Kasson, host of Plantopia. Thanks for listening.